Hello friends, today's my discussion is on properties of cardiac muscle. So properties of cardiac muscle, there are a certain properties like excitability, contractility and conductivity and rhythmicity. So first of all, we will discuss about the excitability. So first of all, what is the excitability? It is defined as ability of the living tissue to give response to a stimulus. And in all the tissues, the initial response to a stimulus is electrical activity in the form of action potential. And this electrical activity that is followed by mechanical activity in the form of contraction or secretion, etc. So electrical potential in cardiac muscle. Resting membrane potential. So single cardiac muscle fiber, it is about minus 85 to minus 95 millivolt, and in SA node it is about minus 55 to minus 60 millivolt, and that is known as pacemaker potential. And in Purkinje fibers it is about minus 90 to minus 100 millivolt. So action potential. Action potential in the cardiac muscle is different from that of the other tissues such as skeletal muscle, smooth muscle and nervous tissue. Duration of the action potential in cardiac muscle, that is about 250 to 350 millisecond or 0.25 to 0.35 second. Then phases of action potential. Action potential in a single cardiac muscle fiber occurs in four phases. Initial depolarization, initial repolarization, then plateau or final depolarization and final repolarization. So this first, that is a depolarization, then second, that is a initial rapid repolarization, and third, that is a plateau, and fourth, that is a final repolarization. So initial depolarization, that is a very rapid process and it lasts for about 2 milliseconds, and amplitude of the depolarization, that is about plus 20 millivolt. Then initial repolarization. Second phase, that is a initial rapid repolarization. So immediately after the depolarization, there is a initial rapid repolarization for a very short period, and it is only about 2 milliseconds. The end of the rapid repolarization, that is represented by a notch. Here you can see there is a slight notch. Then plateau. After that, the muscle fiber that remains in a depolarized state for some time before further repolarization, and it forms a plateau, means stable period in the action potential curve. So this is a plateau phase. This plateau lasts for about 200 milliseconds in atrial muscle fibers and for about 300 milliseconds in ventricular muscle fibers. Due to long plateau in action potential, the contraction time is longer in the cardiac muscle by 5 to 50 times than in the skeletal muscle fiber. Then final repolarization that occurs after the plateau phase. And it is a very slow process and it lasts for about 50 to 80 milliseconds before the re-establishment of the resting membrane potential. Then what are the ionic bases of action potential? So initial depolarization or first phase, that is because of rapid opening of the fast sodium channel and rapid influx of the sodium ion, as in case of skeletal muscle fibers. Then initial repolarization is due to short duration or transient opening of the potassium channel and efflux of a small quantity of the potassium ions from the muscle fiber. Simultaneously, the fast sodium channel close suddenly and slow sodium channel open, that resulting in slow influx of low quantity of sodium ions. Then plateau phase. That is due to slow opening of the calcium channel. And these calcium channels are kept open for a longer period and cause influx of the large number of the calcium ions. Now already the slow sodium channels are open and through which the slow influx of the sodium ion also continues. Because of the entry of the calcium and sodium ion into the muscle fiber, positivity is maintained inside the muscle fiber and producing a prolonged depolarization that results in a plateau phase. Calcium ions entering the muscle fiber that plays a very important role in the contractile process of heart. Then final repolarization, that is due to efflux of the potassium ions. Number of the potassium ions moving out of the muscle fiber that exceeds the number of the calcium ions moving in. So it makes a negativity inside and that results in final repolarization, and potassium efflux continues until the end of repolarization. Then restoration of the resting membrane potential. So at the end of the final repolarization, all the sodium ions which had entered the cell throughout the process of the action potential that move out of the cell, and potassium ion that return back into the cell by activation of the sodium potassium pump. Simultaneously, the excess of the calcium ion which had entered the muscle fiber also move out through the sodium calcium pump, so that the resting membrane potential is restored. Then spread of the action potential through cardiac muscle. Action potential that spreads through the cardiac muscle very rapidly because of presence of gap junction between the cardiac muscle fiber. So in between the two cardiac muscle fibers, there is a gap junctions are present. So because of this, the action potential that transmits very rapidly from one muscle fiber to another cardiac muscle fiber. Now gap junction that are permeable junction and allow the free movement of the ion, and so that the action potential spreads rapidly from one muscle fiber to another muscle fiber. Action potential is transmitted from atria to ventricles through the fibers of the specialized conductive system. Then rhythmicity. So rhythmicity is the ability of the tissue to produce its own impulses regularly, and it is known as autorhythmicity or self-excitation. Property of the rhythmicity is present in all the tissues of the heart. However, the heart has a very specialized excitatory structure from which the discharge of the impulse is very rapid, and this specialized structure is known as pacemaker. And from here the impulses spreads to the other parts through the specialized conductive system. So SA node, that is known as pacemaker, because from the SA node the generation of the impulse is very fast, so that it is known as pacemaker. So what is the pacemaker? So pacemaker is the structure of the heart from which the impulses for the heartbeat are produced, and it is formed by pacemaker cells called P cells. In mammalian heart, the pacemaker is sinoatrial node. It was Louis Sir Thomas who named SA node as a pacemaker of the heart in 1918. Now, SA node is a small strip of the modified cardiac muscle situated in the superior part of lateral wall of the right atrium, just below the opening of the superior vena cava. And fibers of this node do not have contractile element, and these fibers are continuous with the fiber of atrial muscle, so that the impulses from the SA node spread rapidly through the atria. Other parts of the heart such as atrioventricular or AV node, atria, ventricles so can produce the impulses and functions as pacemakers. Still the SA node is known as pacemaker because the rate of the production of the impulse means rhythmicity is more in SA node than in other parts, and it is about 70 to 80 per minute. Then what are the experimental evidences? So experimental evidences to prove that the SA node is a pacemaker in mammalian heart. So first of all, stimulation of the SA node that increases the heart rate. Then destruction of the SA node that causes a immediate stoppage of the heartbeat. And after some time, the atrioventricular node becomes the pacemaker and starts generating its impulses. So the heart starts beating, but the rate is very slow. Then the impulse generates from the SA node. Then local cooling of the SA node decreases the heart rate. Local warming of the SA node that increases the heart rate. And electrical activity starts first in the sinoatrial node. Then spread of impulses from SA node. Mammalian heart has got specialized conductive system by which the impulses from the SA node spreads to the other parts of the heart. So you can see here very well conducting system of heart. So first from the SA node, then comes to AV node, then bundle of His, then left and right bundle branch, then Purkinje fibers, and then to ventricles. So rhythmicity of the different parts of the human heart. In SA node, it is about 70 to 80 per minute. In AV node, 40 to 60 per minute. Atrial muscle, 40 to 60 per minute. Purkinje fibers, 35 to 40 per minute. And ventricular muscle, 20 to 40 per minute. Now pacemaker in amphibian heart. Sinus venosus, that is the pacemaker in the amphibian heart, and it is experimentally proved by the applying a stainless ligature. When the sinus venosus is warmed by a warm ringer solution, the heart rate increases. When the sinus venosus is cooled by a cold ringer solution, the heart rate decreases. The electrical activity starts first in sinus venosus and spread of the impulses from sinus venosus. So amphibian heart does not have any specialized conductive system. Pacemaker in the amphibian heart is the sinus venosus, and impulses from the sinus venosus spreads through the muscles of auricles and ventricles. Then rhythmicity of the different parts of the amphibian heart. In sinus venosus, rhythmicity that is about 40 to 60 per minute, auricular muscle 20 to 40 per minute, ventricular muscle 15 to 20 per minute. Now electrical potential in SA node. So resting membrane potential or pacemaker potential. So pacemaker potential, that is a very unstable resting membrane potential in SA node, and that is also known as pre-potential. Electrical potential in SA node, that is different from that of the other cardiac muscle fibers. In SA node, each impulse triggers the next impulse, and it is mainly due to unstable resting membrane potential. Resting membrane potential in SA node has negativity of about -55 to -60 millivolt, and it is different from the negativity of -85 to -95 millivolt in other cardiac muscle fibers. Now action potential. So depolarization that starts very slowly, and the threshold of -40 millivolt is reached very slowly. After the threshold level, the rapid depolarization occurs up to +5 millivolt, and it is followed by rapid repolarization. Once again, the resting membrane potential becomes unstable and reaches the threshold level very slowly. Then what are the ionic bases of the electrical activity in pacemaker? So resting membrane potential is not stable in SA node. To start with, the sodium ion leak into the pacemaker fibers and cause slow depolarization. Now this slow depolarization that forms the initial part of the pacemaker potential. Then the calcium channel starts opening. So at the beginning there is a slow influx of the calcium ions causing a further depolarization in the same slow rate, and it forms the later part of the pacemaker potential. The initial part of the pacemaker potential is due to slow influx of the sodium ion, but the later part that is due to slow influx of the calcium ions. Then depolarization. When the negativity is decreased to 40 millivolt, which is a threshold level, and the action potential starts with rapid depolarization. The depolarization occurs because of the influx of the more calcium ions. Unlike in other tissue, the depolarization in SA node is mainly due to influx of the calcium ions rather than the sodium ions. Then repolarization. After the rapid depolarization, repolarization starts, and it is due to efflux of the potassium ions from the pacemaker fibers. Now potassium channels that remains open for a longer time and causing a efflux of the more potassium ions. So it leads to development of the more negativity beyond the level of the resting membrane potential, and it exists only for a short period. Then the slow depolarization starts once again, leading to development of the pacemaker potential, which triggers the next action potential. Then conductivity. Human heart has a specialized conductive system through which the impulse from the SA node are transmitted to all other parts of the heart. So conductive system of the heart that is formed by modified cardiac muscle fibers. And these fibers are specialized cells which conduct the impulse rapidly from the SA node to ventricles. And conductive tissue of the heart are also known as junctional tissues. So components of the conductive system in the human heart: AV node, then bundle of His, right and left bundle branch, and Purkinje fibers. So this is a SA node, this is a AV node, then this is a bundle of His, then right and left bundle branch, then these are the Purkinje fibers. Okay, these are the Purkinje fibers. So you can see here the animated conductive system, yellow portion, yellow color that describe the conductive system of heart. So SA node that is situated in the right atrium just below the opening of the superior vena cava. So this is a SA node that is situated in right atrium just below the opening of the superior vena cava. Now AV node that is situated right posterior portion of the intraatrial septum, that is a AV node. Now impulses from the SA node are conducted throughout the right and left atria. So from the SA node, impulses are passes to the left atrium and right atrium. Now impulses also reach the AV node via some specialized fibers called internodal fibers. There are three types of the internodal fibers. Anterior internodal fibers of Beckman. So this is a anterior internodal fibers, means Beckman's bundle. Then middle internodal fibers of Wenkebeck. So this is a middle, that is a Wenkebeck, and this is a posterior. So posterior internodal fibers of Thorel. So this is a SA node, then AV node. So from SA node to AV node, impulse is conducted by three pathways: anterior, middle and posterior internodal tract. And from the AV node, bundle of His and right and left bundle branch, and then Purkinje fibers. All these fibers from the SA node converge on AV node and interdigitate with fibers of AV node. From the AV node, the bundle of the His arises, and it divides into right and left bundle branch, which run on either side of the interventricular septum. From each branch of the bundle of His, many Purkinje fibers arise and spread all over the ventricular myocardium. So this is a SA node, AV node, and these are the internodal pathways. And this is a bundle of His, this is a branch of bundle of His, and these are the Purkinje fibers. Then velocity of the impulses at different parts of the conductive system. Atrial muscle fibers 0.3 meter per second, internodal fibers 1 meter per second, AV node 0.05 meter per second, and bundle of His 0.12 meter per second. And in Purkinje fibers 4 meter per second, and ventricular muscle fibers 0.5 meter per second. So the velocity of the impulse is maximum in Purkinje fibers and minimum at AV node. Now contractility. Another property is contractility. Contractility is the ability of the tissue to shorten in length or contraction after receiving a stimulus. Various factors that affect the contractile properties of cardiac muscle. So all or non law. According to all or non law, when a stimulus is applied, whatever may be the strength of stimulus, the whole cardiac muscle that gives a maximum response or it does not give any response at all. Below the threshold level, if the strength of the stimulus is not adequate, the muscle does not give response. Now all or non law is demonstrated in quiet heart of frog. Heart is made quiescent by applying the first tenuous ligature in between sinus venosus and right auricle. Ventricle is stimulated by placing electrode at the base of ventricle. First one stimulus is applied with a minimum strength of 1 volt at the base of ventricle and contraction is recorded. Then after 20 seconds, the strength of the stimulus is increased to 2 volt and stimulus is applied. And then the curve is recorded. The procedure is repeated by increasing the strength every time and applying the stimulus with an interval of about 20 seconds. Amplitude of all the contraction remains same, irrespective of increasing the strength of stimulus. This shows that the cardiac muscle obeys the all or non law. Then what are the causes of all or non law? All or non law is applicable to whole cardiac muscle because of the presence of syncytium. And in case of the skeletal muscle, all or non law is applicable only to a single muscle fiber. Then staircase phenomena. When the ventricle of the quiescent heart of the frog is stimulated at a short interval of about 2 seconds, here 2 seconds, without changing the strength, the force of the contraction increases gradually for the first few contractions and then it remains same. Gradual increase in the force of the contraction is known as staircase phenomena. So first for few contraction, the amplitude of the contraction increases but then it remains same. So this one is known as staircase phenomena. Then what are the causes of the staircase phenomena? So staircase phenomena that occurs because of beneficial effect which facilitates the force of the successive contraction. So there is a gradual increase in force of contraction. Then summation of subliminal stimuli. When the stimulus with a subliminal strength is applied, the quiescent heart does not show any response. When the few stimuli with same subliminal strength are applied in succession, the heart shows a response by contraction due to summation of stimuli. Then refractory period. Refractory period is the period in which the muscle does not show any response to a stimulus. There are two types of refractory period, absolute refractory period and relative refractory period. Absolute refractory period is the period during which the muscle does not show any response at all, whatever may be the strength of stimulus. It is because the depolarization occurs during this period. So the second depolarization is not possible. Then relative refractory period is the period during which the muscle shows the response if the strength of the stimulus is increased to maximum. It is the stage at which the muscle is in a repolarizing state and refractory period in the skeletal muscle. Now in skeletal muscle the refractory period is very short. An absolute refractory period extends during the first half of the latent period measuring about 0.005 seconds. And relative refractory period extends during the second half of the latent period that measures about 0.005 seconds. So total refractory period that is about 0.1 second. Now refractory period in cardiac muscle. So cardiac muscle that has a very long refractory period as compared to skeletal muscle. Absolute refractory period that extends throughout the contraction period of cardiac muscle and its duration that is about 0.27 second, and relative refractory period that extends during the first half of the relaxation period and which is about 0.26 second. So total refractory period that is about 0.53 second. So what is the significance of the long refractory period in cardiac muscle? It has three advantages. Summation of the contraction does not occur, fatigue does not occur, and tetanus does not occur. So because of the long refractory period, cardiac muscle does not fatigue. Then demonstration of the refractory period in the heart. That is demonstrated in the heart of the pithed frog. Refractory period can be recorded in beating heart as well as in quiescent heart. First, the normal cardiogram is recorded with the heart of the pithed frog and the impulses for the heartbeat arises from the sinus venosus. An electrical stimulus is applied by keeping the electrode at the base of ventricle. When the stimulus is applied during the systole, the heart does not show any response. So whenever we give the stimulus during the period of systole, the heart does not give any response. It is because the absolute refractory period extends throughout the systole. So when the stimulus applied during the diastole, the heart contracts because the diastole is a relative refractory period and this contraction of the heart is known as extrasystole. So this is a extrasystole. So stimulus giving during the diastole. So heart contracts because diastole is a relative refractory period and that is known as extrasystole or premature contraction. Extrasystole, that is followed by the stoppage of the heart in diastole for a while, and this diastole is longer than the diastole after the regular contraction. Temporary stoppage of the heart before it starts contracting, that is known as compensatory pause. So this one is a compensatory pause. Duration of the extrasystole and compensatory pause is equivalent to the duration of the two cardiac cycle. Then what are the causes for the compensatory pause? So a natural impulse from the sinus venosus arrives at the time of contraction period of extrasystole. As this period is absolute refractory period, the natural impulse cannot cause the contraction of the heart. And the heart has to wait for the arrival of the next natural impulse from the sinus venosus. So till the arrival of the next impulse, the heart stops in the diastole. Then refractory period in quiescent heart. Frog's heart is made quiescent by applying the first venous ligature. Electrode is placed over the base of the ventricle. When the two stimuli are applied successively in such a way that the second stimulus falls during the contraction period and the heart contracts only once. And it is because of the first stimulus. There is no response to second stimulus because the systole is the absolute refractory period. However, when the second stimulus is applied during the diastole, the heart contracts again and the second contraction superimposes over the first one. And this shows that the relative refractory period extends during the diastole. Thank you very much.